All right, welcome. So, autumn. Yay, it's autumn. I love it. It's my favorite time of year. And it's a really great time of year and a great place here in the Northwest to find a diversity of mushrooms. We were just talking about that before the recording started. So, our next field trip is going to be to Fort Stevens State Park, where we're going to join one of their mushroom walks. And that's the 27th, the last Friday of the month. So while we're there, we'll be able to gather some mushrooms that we can sketch after the program. And mushrooms come in a diverse array of shapes and colors and textures, and they're a fantastic subject for practicing your nature journaling skills. So the purpose of today's session is to show you some of the details that you'll want to record when you're observing mushrooms so that you'll be prepared for our field trip later this month. So fungi are a group of organisms that are so unlike anything else that they've been given their own kingdom in the classification of life. They're similar to plants in how they grow, but their feeding habits are more like animals in that they digest and consume organic matter. There's estimated to be like 2 to 4 million different species of fungi in the world, although just a fraction of those have been described so far. And of those millions of organisms, they're spread across 6 different subkingdoms, which are listed here on the slide. The organisms that we know as mushrooms are actually all in the subkingdom of Dicaria, in the divisions of Basidia mycota and Asca mycota. So just those, you can see it's just a small little fraction of all the fungi in the world actually produce mushrooms. The basic difference between these two groups, the Basidia and the Asca, are the way they reproduce. And so the Basidia mycota, they have spores that form on the outside of their reproductive cells, whereas the Asca the Asca mycota, they have cells that are inside capsules, so they're or sac-like reproductive cells, so they're more protected and enclosed. But besides that, there's similarities between the two groups and vast differences within each of the groups. So the parent body, or the vegetative portion of a mushroom, is known as the mycelium. And the mycelium is actually like a diffuse network of thread-like hyphae that whose tips excrete enzymes that digest organic material, and then the mycelium absorb that juice. When the mycelium, so basically in the GIF here, you can see when the stick is turned over, there's a layer of kind of white mold. That's the mycelium of the mushrooms that you see on the surface. And in the ground, like this picture in the lower right, the mycelium can just look like a spongy white material or like mold because it's a fungus. And under certain conditions, when the mycelium has gathered enough energy and the temperature is just right and the moisture levels are just right, some of those hyphae will form together in a little bundle and start to swell. And that swelling bundle is basically their reproductive organ, and it'll erupt from whatever substrate it's growing in, and that's what produces the mushroom. That is the mushroom itself. And you can see this little diagram here. It's a complex process. They're actually I don't know enough about this to speak of it intelligently, but they reproduce sexually, and so the hyphae split and join, and then resplit and rejoin throughout this process of creating a mushroom, and then creating spores, and then creating new hyphae. And so it's this complex mixing of genetic material that's visually represented in this graph here. So some of the mushrooms are distinct enough that identification of them is pretty straightforward. Like the fly agaric that was mentioned before the recording is a red cap mushroom with white flecks on the surface. It's pretty distinct. But a lot more of our mushroom species are so similar in appearance that making positive identification of them can be pretty tricky. So some of the characteristics that you would want to look at to help identify mushrooms are listed here. I got these from the website that's at the bottom of the list here. We're going to look at a few of these more closely. Some of these, like chemical reactions, that's not something you can do in the field. Odor, I actually haven't ever smelled. I guess I haven't smelled enough mushrooms to know this, but apparently there's a few that you can identify by the smell. But a lot of these other things you can actually collect that data in the field. And so we'll look at a few of those. But here is a general image of your typical stipe and cap mushroom shape and the names of the different parts. And so the flecks on the surface of the cap are remnants of veil. And there's two different kinds of veils that can be present. One is the universal veil, and you see a little sketch of it here. This is the remnant of it. A universal veil, I have a slide with this later, but it encompasses the entire mushroom as it's developing. And then as the mushroom expands, it busts out of that veil. The annulus, this remnant on the stem or the stipe, is it's a remnant of a partial veil, which is one that only covers the cap and the hymenophore or the spore producing area. And then it also will break free as the cap expands. The mycelium are quote unquote the roots, but they're actually the entire organism. And let's see, yeah, that was everybody. And the vulva is the official term for the remnant, the base remnant of the universal veil. All right. So your overall shape's the first thing you're going to notice, and it can give you a good starting place on identifying or keying out your mushroom. So how might you describe some of the shapes that you're seeing here if you were to have to write them down in your journal? You can use the chat or just say it aloud.
Some I'd say are coral-like,
Okay, coral-like.
shelf, or they have a shelf, or they're flat and stick out like a shelf.
Okay.
The one in the center bottom I would describe as having tendrils.
Okay.
And actually the one in the center on the top maybe lobed. Sort of looks like the brain matter, kind of.
Yeah, I like that. And in the chat we have finger-like. Most of the mushrooms that we see tend to have this typical appearance, but like the toadstool shape with the cap and the stipe. But there are others out there, and all of these were taken locally, and these are all different shapes that you might see when we're on our field trip. All right, so when I was looking for official names of different shapes, I found this website and I wanted to share it with you. It's not, it doesn't just cover fungi that are in the Dicaria group. So there are some images that are in this that are not mushroom producing, but it's kind of a fun website to look at. So if you want to see, basically it's just keying out fungi that grow on wood. And there's a long list of these different shapes, and then it has a key, or you can go straight to the species list and then look through the species list and see all sorts of different kinds of fungi. So that's kind of fun. In fact, I can put oh no, I can't access that right now. Okay, I was going to put it in the chat, but I can share that with you later. All right, so the next thing to look at is the shape of the cap, its color and its texture. So in addition to coming in a variety of colors, as we were talking about earlier, the caps can come in all sorts of different shapes and textures. And these are all just regular, like quote unquote regular toadstool shaped mushrooms, but you can see that there's different textures on each one and some the edges are different on some. So what are some words you might use in your journal to describe the differences that you see here? Like how might you describe this brown one here as like how would you describe its texture? Or this one here, or this one here, or this one or this one.
Which one is the brown one?
Oh, upper left, upper left.
I would call the surface of that, at least from what I can tell here, kind of it looks velvety. It looks very soft,
Mm-hmm. Yeah.
a little fuzzy.
Another term I've heard for that I read today was like suede. Yeah. Velvety was the term that was coming to mind for me too. And then like suede, I was like, oh yeah, that's a good one too. How about here in the lower right corner?
That one looks very moist and sticky potentially.
Mm-hmm. Yeah, some of them will look wet even if it hasn't been raining, which I find fascinating.
I
And
was
then
gonna I was gonna
oh,
say
go ahead.
it has a sheen.
Yeah, it looks lacquered or wet or yeah, sheen. It's hard to tell with this one in the upper center, but the surface of that one is kind of scaly. And in the lower left side, this is also kind of a velvety, soft or matte texture. Um, yeah, so we'll see a lot of different, and these are, for me anyway, I have a difficult time capturing details like this in my journal because they're so, in my sketching, because they require a level of skill that I haven't acquired yet. And so looking for words to describe these characteristics helps me a lot in capturing that when I am drawing an image of it. So even within your standard mushroom shape, the toadstool shape, there can be a wide variety of cap shapes between species. If you look up mushroom cap shapes online or in your field guide, in your mushroom field guide, there's some official names for them, and those are some of the ones I shared here. But you can also just come up with your own names as you're journaling and to help you describe them. But when the center is lower than the edges, it's called depressed. When the edges are lower than the center, that's convex. Sometimes that convex shape is very conical or it can be round, and sometimes the depressed shape can be quite exaggerated, and then that is called a funnel shape. And so with these mushrooms, if you were to look at them from the top, they're like vases. The there's the depression in the center goes way down the stipe here. So some more characteristics to look at. And yeah, so another thing about the caps to notice is if they have these remnants of the veil. So here's an example of a mushroom that's just starting to break out of its universal veil. So you see that it almost looked like there was an egg in the ground that's starting to hatch, and the mushroom will come up and then there'll be that vulva down at the bottom, which can be hard to find sometimes because it could break below the surface. And then sometimes it'll leave little flecks on the cap, like you see in the center image, or it can leave lots of flecks on the cap, like the two images on the right. The partial veil here, you can see that it's still closed on this mushroom, and it's as if the cap is wrapped around tightly. The bottom of the cap is wrapped tightly around the stem, and then as that cap expands, that partial veil will tear and rip free. And sometimes there'll be remnants on the edges of the caps, and most often the time there'll be remnants around the edge of the stipe. So those are other details to capture because those can help differentiate between one species and another. It is important when you're learning a mushroom. Actually, this is in the next section, but Actually, I'll just go to the next section. I'll talk about it later. Okay, so the next thing to look at on the cap is the spore surface, which is on the underside of the cap. And the spore surface can really help you quickly identify particular edible mushrooms, for example. So if you're looking at an orange mushroom and you're wondering if it's a chanterelle, if you look at the underside and you see these ridges, then you know it's a chanterelle. Although I did just learn today that there's a false chanterelle that has veins or gills that look very much like ridges. So it's good, like we were talking about before the recording started, to be very familiar with the mushrooms that you're picking before you decide to eat them. Hedgehog mushrooms are another edible. They always have teeth. And boletes, not all the boletes are edible, but all boletes have pores. So gills is the term for the typical spore producing surface that you see on mushrooms that you buy at the grocery store. For example, those little white button shaped mushrooms, they have gills, and they're like freestanding sheets of material that radiate outward from the stem. They're very easy to tear off. Ridges also radiate from the stem to the outer edge of the cap, but they interweave and they are more like just raised surfaces on the underside of the cap as opposed to almost freestanding surfaces like the gills. Pores look like thousands of tiny holes have been poked in the underside of a solid cap, and teeth look like thousands of stalactites hanging down from the cap surface. So these are very distinct spore producing areas. So the hymenophore, or the spore producing part of the mushroom, can help you distinguish between edible and its lookalike. And so here are a couple of mushrooms from the region around where I'm at. The one on the left is a chanterelle, and the one on the right I haven't identified yet, but it often pretends to be a chanterelle. So when I'm chanterelle hunting, it grows in the same type of habitat. It has similar grouping patterns. Chanterelles will often grow in groups, and that it's a very similar color, and sometimes it has an uneven cap like the chanterelle. So it can be it can trick me sometimes until I flip it over, and it has gills instead of ridges, and you can see here that the difference is quite distinct, and the gills are a different color. The ridges on a chanterelle tend to be lighter, and on this mystery wannabe chanterelle they're darker. So that's when I get tricked, that's how I tell that it's the wrong kind. So like the caps, stems can come in a range of colors, shapes, and textures, and recording and noting these features can help with identification. This is a resource I found online. You can see the website here at the bottom, and some pictures of local mushrooms. And the morel down in the lower right isn't local to me, but I put that in there because this is another case where understanding the characteristics of the mushroom that your target species is will help you differentiate it from poisonous or unwanted lookalikes. So in the case of the morel, if you look at these three characteristics in the lower right-hand corner of the diagram, you have solid, hollow, and chambered. A morel mushroom has a completely hollow interior from the cap all the way to the ground. It has a lookalike, a toxic lookalike that has a chambered interior. And so when you cut it open, they're sectioned like bamboo, right? As opposed to being one long solid hole or one long opening. And I'm not that familiar with this kind of mushroom. And when I look at the two species together, I couldn't tell them apart. But looking at the interiors, if you slice it vertically and look at the inside, that would be a characteristic to help tell the two apart. So just like the caps, the stems have remnants of partial and universal veils. And so here are some examples of the universal veil. It's called the vulva when it's at the base like this. And then here are a couple examples of the partial veil and the remnant on the stipe. Something I was going to say earlier is that when you're learning a new mushroom, you will want to dig it up a bit so that you can see if there is a universal veil under the ground, because sometimes it isn't above the surface. And the presence or absence of a universal veil can be the difference between a toxic and a non-toxic mushroom. And so it's important that you really get to know this organism from underground to the top before you decide to eat it. Another thing when learning about mushrooms is it's really helpful to know the time of year where the mushroom, or to note the time of year where the mushroom's growing and what it's growing from, if you can tell. So some species only fruit in the spring, others in the fall. Some like evergreen forests, others like hardwoods. Some will grow anywhere, and lots will grow directly from dead wood, as you can see in some of these pictures. But some within that category, these would be the saprophytic, growing from dead organic material. Some will grow on wood that's above ground, like still standing trees. Others will grow on wood that's on the ground, but still relatively solid. And others will grow on wood that's more decomposed. And so noting these little details can help you to learn more about the organism that you're finding. Others can be, this one's growing from a tree here, but these ones are growing, it looks like, from the ground. And so it can be difficult to tell if they're actually growing from the soil or if there's some dead material underneath that they're eating. And so this is another reason that kind of digging them up a little bit can give you some more information about them. This image on the left is someone harvesting morels, and that was in May. And then here is someone harvesting a cauliflower mushroom, which comes out in the fall. And so this is a picture taken in November. Let's see. Oh, and also, so paying attention to where the mushroom's growing and what it's growing on can help you learn about the mushroom. But if you're familiar with the mushroom and its feeding strategies, that can tell you information about what's happening below the surface. So that's kind of fun too. So the knowledge, they can teach you more about the environment they're living in, and you can learn more about them. So feeding strategies, speaking of what the mushrooms are growing on, there's 3 general feeding strategies in the fungal world. One of them is parasitic, and this is when it's growing on a living organism. We don't have any, well, I don't know. I'm not familiar with any parasitic mushrooms in our region. An example of a parasitic mushroom would be cordyceps, which is, it grows out of an insect, but a lot of the parasitic fungi don't produce mushrooms in order to reproduce. Now, we do have a parasitic fungus that grows on mushrooms in our region, and it is referred to as the lobster mushroom. And so it'll take, I think it's a russula, it'll grow on the surface of the russula and transform it into this hard orange surface. And apparently they're edible, but I'm afraid to eat them because I don't understand what's going on there, and I can't be 100% sure what they grew on because they distort the surface so much that it's, for me, not recognizable. So I like looking at them, but I don't eat them, but I know people who do. The next feeding strategy is saprophytic, and I think, or saprophytic, and that, in my experience, I think that might be, well, it's the one I'm most familiar with seeing out in the forest. Now, the obvious ones are the ones that are growing straight out of trees, like the conchs and the other polypores. But there's also a lot that grow, like we saw some images of some growing in woody debris on the ground as well. And then the third feeding strategy is the mycorrhizal fungi. And these are the organisms that form symbiotic relationships with plants, usually trees. So in these relationships, the plants are providing moisture and organic compounds like carbohydrates to the fungus. And the fungus is helping the tree access nutrients like phosphorus and nitrogen. Some saprophytic fungi can function with a variety of hosts, like this violet court. This violet court can form symbiotic relationships with a variety of different tree species. But other mycorrhizal, I said saprophytic, I meant mycorrhizal. So some mycorrhizal fungi function with a variety of hosts. Others are specific to a particular species. And so when you're learning your mushrooms and making a note of who is in the area around the mushroom that you're finding is another way to kind of learn more about who they might be collaborating with, and also help you if you want to find them again in the future. So you can see this little guy, there's a hemlock growing in the background. So it's possible that, especially since there's so many hemlocks growing in this area, that that might be the tree that this particular fungus is collaborating with. So those were all characteristics that you can do in the field. Something that you can do at home is to take a spore print. So to do a spore print, you basically just need the cap of a mushroom, a surface to receive the spores, and something to put over the top so that the breezes don't blow through in between the cap and the paper. Spores are very tiny, and any kind of breeze will create kind of what you see in this image on the right, which is my attempt at a chanterelle, and I didn't do it correctly. And so there's a little bit of detail in the center, but you can see there's also a lot of smudging. Spores come in a variety of colors, and so if you have the opportunity to do your spore print on something transparent, then you can put different color backgrounds depending on what the spore print ends up being. So a chanterelle spore print is kind of this really light peachy color, and so I did it on black paper, but I already knew that beforehand. You don't necessarily know by looking at the mushroom what color the spore print is going to be. And so if you're learning something new, it can be helpful to do it on something transparent. Let's see. When you're doing a spore print, what I want to say about this, it's best to use a freshly mature cap. Older caps will be less, give you a less clear image. When you do a spore print, you want to leave it as long as possible. I would say at least 24 hours. And then after you do the spore print, when you're finished letting the spores settle, remove the cap and then let that surface dry. If you try and process it right away, it could get moldy if it's damp. So let the spores dry, and then after they've dried, I haven't tried this technique myself, so I can't I can't attest to how well it works. But what I've heard is that spraying it with hairspray or artist's fixative will help keep that spore print in place. And you want to make sure though when you're doing it that you're holding the spray at least a foot away so that you don't blow the spores off of the page. Oh man, sorry about that. I just wanted to do this. Just checking my notes to see if there's anything I left. Oh, and if you're going to take a spore print and you want to put it in your journal, remember to leave space in your journal for the print. Yeah, when I did this print on the chanterelle with the chanterelle, part of the reason that it blurred is I did it on black paper, and then to adhere it into my journal, I slipped it into a piece of that clear plastic stuff that comes on the outside of greeting cards sometimes, what that's called. And it can move around a little bit, I think, and so that also smeared the spores. So I won't be using that technique in the future. I'll be trying the artist fixative next time. Has anybody else had tried doing spore prints before and putting them in their journal?
I've done spore prints quite a bit, actually.
Okay. Do you have any tips for us?
When we were in Alaska, we tried the hairspray to fix it.
Yeah.
Because nobody brought fixative. So it helps. It kept them good until I got home, but it's not a very secure method.
Okay.
I could still rub it off if I like put my finger on it and I moved my finger by accident or whatever. But if it was just sitting in my journal and not being disturbed, it held the spores pretty much in place until I got home. But I would use an artist fixative. It works a lot better to keep it in place. So that's one thing. Like I said, it did work in a pinch to at least get it from point A to point B. But yeah, I actually love doing this. This is the funnest thing ever.
Yeah. I've only done it a few times myself, but I love the results. I love the results. Another thing that I was thinking about trying, but I haven't yet, so I don't know if it would work, is to take the spore print, let it dry, and then use some of that clear packing tape and put the clear packing tape over the surface. But it's possible that, you know how it's pretty staticky, and so it's possible that putting the tape down towards spores will cause the spores to leap off the page and get all mushy. So I don't know if that works yet either. It's something that I'll learn this fall as I practice. But
so somebody was suggesting that as well as a secure method. I would probably spray it a little bit first, like even if it's just a really light spray to keep that static down.
Yeah.
My fear is that for the long term, I'm not sure how secure packing tape would be.
Okay.
I mean, because you look at old photo albums or whatever where people taped in picture or whatever, and the tape's now not sticky anymore. It's kind of gone away. Even, from when I moved, some of those packing boxes, the packing tape is not very secure anymore. So I'm not sure. I mean, I love the idea because what better way to secure it in place? You can do anything with it. But I'm not sure if it would be considered archival or long-term.
Right.
But
That's
maybe,
a good point.
I don't know. But I wanted to try it as well because I think it would be more secure in a journal that's getting, pages are getting turned. It's getting looked at a lot. I wonder if there is some kind of like for scrapbooking or some kind of art product that is more archival and more secure than like just going down and buying, shipping tape.
Yeah. I bet you there is. To look into it
that would
more.
be my only suggestion. And like I said, I haven't tried that method, but I want to. But I was going to do some research and what I thought might work best for the longest haul.
Yeah. Good points. I hadn't thought about that, about the lack of longevity of the packing tape. It's a really good point. Yeah. Cool. Awesome. All right. And does anyone have the time? I apologize. I don't have a clock on me. How are we doing?
7:45.
Oh, okay. Fantastic. Thank you. All right. So to recap, here are some of the details that you want to capture about your mushrooms. This is just me. Obviously, you do a spread however it makes sense to you, but you want to catch the date, the time, the weather, and location, your basic metadata, and then a sketch of the whole mushroom with notes about the texture, the color, and the size of the different parts, and a sketch or description of the spore-bearing surface, the underside of the cap, a description of the habitat and the substrate where your mushroom was found. And then if you're going to do spore prints, leave a little spot somewhere for a spore print. If you've got a huge mushroom, you can do a partial print. You don't have to do the whole thing. Like if it's not going to fit on a journal page, you could just do take a spore print of like a quarter or an eighth of the cap and stick that in if it'll fit. So let's see. Oh, equipment. Yes. So we're going to have a field trip later this month. In the book Mushrooms Demystified, David Aurora suggests the following equipment for your mushroom forays: a rigid container, something that won't crush your mushrooms. If you don't have a rigid container, at least don't use a plastic bag. The problem with plastic bags is that they trap moisture And that will cause your mushrooms to get slimy really quick. I have experienced this. I can attest to that. It's gross. Um, if you're going to collect mushrooms, and some of them you might eat later, make sure that you keep all your different specimens separate from each other. You can use wax paper or small paper bags, but you don't want mystery mushrooms touching your edible mushrooms, because you don't know if those mystery mushrooms are toxic or not, and they might, you know, I don't know, they might transfer. Well, I think the big concern is that you might accidentally imbibe, eat one of those toxic ones if everybody's all mixed together. So if you're going to eat any of them, keep everybody separate. And a knife or a trowel, something for cutting or digging up mushrooms, your journal and your writing utensil for recording your observations. Binoculars are something to consider. I have used them myself when mushrooming. The obvious thing is, like, when you're mushrooming, you're also experiencing other aspects of nature. And so if you have your binoculars on, you can look at the birds who are migrating through. But they can be helpful with mushrooming. So if you're hunting for a specific specimen, for example, and you're in a really hilly area with valleys and steep slopes, and you see a bit of color on the opposite hillside, you can use your binoculars to tell whether or not that's a leaf or a chanterelle, for example, from personal experience. And because sometimes the leaves look just like chanterelles. What else would be? Oh, hand lens. So if you want to look at something more closely, bring a hand lens or a magnifier with you. If it's dry out, a field guide, if you like keying things out in the field, rain gear, because it probably will be wet, and a camera, which is a great data collection tool in itself. A note about rain gear: I have experienced that sometimes I get more wet if I'm wearing rain gear because I'm hiking around so much, and the rain gear holds the sweat in. And so just something to think about. If you're wearing rain gear, breathable rain gear is preferable, because it can be a bit humid and warm even if it's raining out here on the coast. So I thought, oh, I guess I should wrap up. So mushroom hunting is a great excuse to get outside when the gray days and rainy weather make you want to stay inside. And by paying attention to where they grow, like what they're emerging from and who they're growing with, you can tell, uh, you can learn more about the landscape that you're exploring. They add this additional layer of data that makes your nature experiences richer. So I, I encourage you to go mushroom hunting, even if you're not able to come on the field trip with us at the end of the month. Just going out and exploring and seeing what you find can be incredibly satisfying.